Hello everyone, I am Adebola Orimadigun. I'll be talking about the life cycle of malaria parasite and severe malaria in this lecture. So in this lecture, I will expect that all participants will be able to describe the life cycle of malaria parasite at the end. Everyone should be able to explain the pathophysiology of the various manifestations of severe falciparum malaria and to be able to explain some of the relevance of the pathophysiological mechanism to clinical practice. Malaria is a febrile illness caused by the protozoa of the genus Plasmodium. The human malaria is caused by the following Plasmodium species: Plasmodium vivax, Plasmodium falciparum, Plasmodium ovale, Plasmodium malariae, and Plasmodium norsale. Of the 5 species of malaria, the most fatal is the Plasmodium falciparum malaria. It is responsible for most cases of severe malaria in sub-Saharan Africa, also in Asia, but vivax is more common than falciparum in Asia continent. Plasmodium falciparum is the commonest species in Africa, and it is said to be accounting for about 96 to 98% of cases. Plasmodium vivax and ovale have the dominant liver stage parasite, otherwise known as the hypnozoite, which can be reactivated and cause relapsing malaria after several months or years. Plasmodium malariae produces long-lasting infection and, if left untreated, can persist symptomatically in human hosts for several years as well, even for a lifetime. Plasmodium norsale is a zoonotic infection. It's seen mostly in the forest region of Southeast Asia. These diagrams show the areas of the world where malaria persists, the cases and deaths in Africa. Malaria is most common in central part of African region, both north and south of the equator. Africa, the southern part of sub-Sahara, and portion of Oceania such as Papua New Guinea have the highest transmission rate. According to the latest World Malaria Report, there were 241 million cases of malaria in 2020 compared to 227 million cases in 2019. The estimated number of deaths stood at 627,000 in 2020, an increase of 669,000 deaths over the previous year. While about two-thirds of these deaths were due to disruption during COVID-19, the remaining one-third of the deaths reflected a recent change in WHO methodology of calculating malaria mortality, irrespective of COVID-19 disruption. The African region continues to bear disproportionately large amount of the world malaria burden. In the year 2020, the African region accounted for 95% of all malaria cases and 96% of all deaths. Children under the age of 5 years accounted for around 80% of the malaria deaths in the region. Nigeria, the Democratic Republic of Congo, the United Republic of Tanzania, and Mozambique accounted for slightly more than half of all deaths globally. All these total 3.8%. And as you can see, Nigeria 31.9, DRC 13.2, Tanzania 4.1, and Mozambique also 3.8. The life cycle of malaria parasite. Human malaria parasites undergo a complex cycle of development, alternating between two hosts, the mosquito vector and then the man as the host. The life cycle of malaria parasite occurs in a sequence of phases, which include sexual phase in the mosquito and asexual phases in man. The asexual phases in man comprise erythrocytic and exoerythrocytic cycles, as you can see in the diagram. During the stages of malaria life cycle in man, the exoerythrocytic cycle begins when human hosts get infected as the female Anopheles mosquito injects saliva containing tiny elongated sporozoids into the bloodstream during its blood meal. You can see that in the figure. Within an hour, these sporozoids are carried by the bloodstream to the liver, where they invade the parenchymal cells, initiating the pre-erythrocytic cycle, otherwise known as exoerythrocytic cycle. In the hepatocyte, the sporozoid undergoes changes in the morphology, losing their apical complex and their surface coat, then transform into a round or oval trovozoid. Development takes place within a parasitophorous vacuole in the hepatocyte. The trovozoid increases in size and finally develops into a tissue rhizome, also known as marrow, containing several thousands of merozoites. These get distended and then the cell ruptures, releasing merozoites into the bloodstream. And this marks the beginning of erythrocytic cycle within man. The erythrocytic cycle simply refers to the life cycle of the parasite within the red cell. This begins by merozoites getting attached and invading the erythrocyte, and then the cycle begins. They have distinct features and are specialized uh, in recognizing the red cell and the specific molecule in particular in the membrane make this happen. After invading the erythrocyte, the parasite loses its specific invasion organelle and differentiates into round trovozoids located within a parasitophorous vacuole in the red cell cytoplasm. Within the red blood cell, the trovozoid is vacuolated and looks like ring in shape, more or less amoeboid and uninucleate. The young trovozoid grows substantially before undergoing several nuclear division. This soon then follow the division of cytoplasm, forming a rhizome, and each containing merozoids in number characteristic of the parasite species. The merozoids then invade fresh erythrocytes, and the cycle continues as you can see in the diagram. The length of this erythrocytic cycle depends on the type of the parasite species. For Plasmodium vivax and ovale, the cycle may take approximately 36 to 48 hours, and that is why infection by these two species are described as tertian. For Plasmodium falciparum, the periodicity is still defined, but it's also within that range of time. For Plasmodium malariae, it takes about 72 hours, and that is why the malaria caused by P. malariae is described as cotton malaria. After a few erythrocytic cycles, some merozoids develop into male and female gametes, but whether this is determined by genetic makeup of the parasite or as a result of specific immune stimulus remains unknown. However, it's important for us to note that the erythrocytic cycle is of clinical relevance because it is during this stage that certain chemokines are released from as a result of the ruptured red cell and then initiate some of the pathophysiology and pathological features that we shall describe later on. The stage of malaria life cycle in the mosquito begins after the female Anopheles mosquito takes up the gametocyte. Now, looking at this diagram closely, the gametocyte that is taken up by the mosquito are transformed into gametes, and the maceration of the female gametocyte into macrogamete takes place without major morphological changes. In the extracellular male gamete, the nucleus divides 3 times, and each of the 8 nuclei formed combined with cytoplasm to form 6 to 8 thread-like microgametes, which is X-flagellate. The sporogony phase, which you are seeing in this diagram, occurs within the mosquito, and that begins with fertilization between the microgamete and the macrogamete, which occur within the stomach wall of the mosquito, resulting in the formation of what is known as diploid zygote. The diploid zygote gets elongated quickly and become a mobile eukaryote, and which slowly moves across the peritrophic membrane of the midgut, and then passes through the single cell layer of the midgut epithelium of the mosquito and establishes itself within the basal lamina, forming an oocyst. The oocyst gradually increases in size and then enlarges and undergoes nuclear division, forming sporoblast. The sporoblast then in turn divides and then repeatedly to form thousands of sporozoids, and then being motile, the sporozoids burst through the weakened and ruptured wall of the oocyst and then invade the body cavity of the mosquito. From there, it travels into the lymph sporozoids, the lymph, and then migrates into the salivary gland. Then from the salivary gland, the sporozoid is ready to be injected into man again the next time the mosquito takes its blood meal. Let us discuss pathophysiology of malaria fever and other features. Malaria is caused by the infection of red blood cells by the asexual forms of malaria parasite, and the involvement of red blood cells makes malaria potentially a multi-system disease because the blood reaches every organ of the body. Malaria manifests with usually fever. However, some people may proceed to have other features, including life-threatening features, which typifies severe malaria. At the end of the schizogony within the red blood cell, that is the erythrocytic cycle, each cycle lasting 24 to 72 hours, depending on the species of the infected parasite. A freshly formed merocytes are released by lysis of the infected red cells, along with numerous known and unknown red blood cell products. Some of these products include cell membrane components, hemozoin pigments, and other harmful factors, such as glycosyl phosphatidyl inositol, otherwise known as GPI. These substances, particularly the GPI, activate macrophages and endothelial cells, causing them to secrete cytokines and inflammatory mediators such as tumor necrosis factor, interferon, interleukin 1, interleukin 6, interleukin 8, macrophage colony stimulating factor, and lymphotoxin, as well as superoxide and nitric oxide. Many studies have suggested that the GPI tail, which is found on multiple merozoid surface protein, including MSP1, MSP2, and MSP4. MSP simply means merozoid surface protein. These substances have significant parasite toxic effect. The various cytokines released in response to these parasites and red cell membrane products have been largely attributed to the systemic manifestations of malaria, including headache, fever, rigors, nausea, and vomiting, diarrhea, anorexia, tiredness, joint ache, muscle ache, thrombocytopenia, immunosuppression, coagulopathy, and central nervous system manifestations. In addition to these features, Plasmodium DNA is extremely pro-inflammatory,
and this can cause elevated cytokine levels as well, and as well as fever. The plasmodial DNA is presented by hemoglobin, which is produced during the parasite development within the red cell to interact intracellularly with toll-like receptor 9, resulting in the release of pro-inflammatory cytokines, which in turn induce COX-2, regulating prostaglandin, resulting in fever induction, and then hemoglobin has also been related to apoptosis, a growing erythroid cell in the bone marrow, and this also results in anemia.
It is important to note that many and several systemic manifestations of malaria are attributable to the cytokines released, and the list is endless. For the purpose of focusing malaria management, WHO recommends that malaria be classified into uncomplicated and complicated malaria. The uncomplicated malaria simply imply that the symptoms of the infection are without evidence of vital organ dysfunction or life-threatening clinical symptoms and signs. So most cases of malaria in children in the tropics belong to this group of category of clinical malaria, and they are straightforward, uncomplicated malaria that require treatment. Now, the other uh, aspect of malaria is the severe or complicated malaria. This is described as presence of evidence of vital organs dysfunction.
The life-threatening features of malaria, which characterize severe malaria, was first defined by WHO in 1990, and the list is as shown on this slide and the next one. First, cerebral malaria is defined as unrousable coma not attributable to any other cause, with a Glasgow coma score of less or equal 9 or Blantyre coma score of less than 3. Coma should persist for at least 30 minutes after a generalized convulsion. Severe anemia, which is another form of severe malaria, is defined as hematocrit of less than 15% or hemoglobin of less than 50 gram per liter in the presence of parasite count of greater than 10,000 microliter. Renal failure is another feature, which is defined as urinary output of less than 400 ml in a day in adults and less than 12 mils per kilogram per day in a child, plus a serum creatinine level of greater than 265 micromole per liter or greater than 3.0 milligram per deciliter, despite adequate volume depletion. In other words, you must be sure that the child is not hypovolemic, not dehydrated, and adequate fluid therapy has been administered. Pulmonary edema and acute respiratory distress. The acute lung injury score should be used in this case and should be based on radiographic density, severity of hypoxemia, and positive end expiratory pressure. Hypoglycemia is another feature of life-threatening malaria, and it is defined as whole blood glucose level of less than 2.2 millimole per liter or less than 40 milligram per liter. Another feature is circulatory collapse, otherwise known as acute malaria. This is defined as systolic blood pressure below 70 millimeter of mercury in children above 5 years of age, or systolic blood pressure of less than 50 millimeter of mercury in those aged 1 to 5 years with cold climate skin or core temperature difference of greater than 10 degrees Celsius. Another feature is abnormal bleeding or disseminated intravascular coagulation. It is defined as spontaneous bleeding from GOB, notes, GIT, or laboratory evidence of differentiated intravascular coagulation. Other features are repeated generalized seizure of 3 times month within 24 hours, plus, of course, P. falciparum parasitemia. Acidemia or acidosis is defined as arterial pH of less than 7.25, or acidosis, defined by carbonates of less than 15 millimole per liter. Macroscopic hemoglobinuria is another feature of severe malaria. This is defined as hemolysis that is not secondary to G6PD deficiency, that is glucose 6 phosphate dehydrogenase deficiency. After this list, in the year 2000, WHO added some more criteria, and these include impaired consciousness, rousable mental condition, but alteration in the level of consciousness, prostration or weakness, which is defined as inability of a child to drink spontaneously or sit uprightly if the child could previously do so. Hyperparasitemia is another feature and is defined as parasitized erythrocyte of greater than 5%, or parasite count of greater than 250,000 parasites per microliter of blood in non-immune individuals. Hyperparexia is another feature of severe malaria. It's defined as core body temperature of greater than 40. And the last one is hyperbilirubinemia, which is defined as total serum bilirubin of more than 43 microliter per micromole per liter. I beg your pardon. Or greater than 2.5 milligram per DL of bilirubin.
Before we go on, I would like to make clarification on clinical definition of cerebral malaria. Please note that it is a form of severe falciparum malaria with cerebral manifestation, which is characterized by plasmodium falciparum parasitemia, unarousable coma, which depicts Plancos coma scale of less than 11 or Blantyre coma scale of less than 3, and absence of other causes of coma. So you must rule out other causes of coma before you make a diagnosis of cerebral malaria. Also, please note that convulsion associated with loss of consciousness persisting for more than 30 minutes after a seizure with P. falciparum malaria demonstrated in blood film could be considered as cerebral malaria. So you don't have to wait on the patient develop frank coma. Now let's look at pathogenesis of severe malaria. The pathophysiological factors involved in development of severe malaria include parasite biomass, malaria toxin and inflammatory responses, cytoadherences, resetting and sequestration, altered deformability and fragility of parasitized red cell, endothelial activation, dysfunction and injury, and also altered thrombosis, which affects coagulation. The role of biomass in pathogenesis of severe malaria have been well highlighted in literature. P. falciparum is known to have ability to invade red cell of all ages, with repeated life cycle of development within the red cell leading to heavy parasitemia. P. vivax, on the other hand, preferentially infects only young red cells, thus limiting its reproductive capability and the resultant parasite load. P. falciparum can infect as many as 20 to 30% of the red cell, but P. vivax could only infect just about 2%, and that may explain the rarity of the severe forms of P. vivax malaria in Africa. Now, the role of cytokine in severe malaria, it is known that excessive level of cytokines leads to decreased mitochondrial oxygen use and enhanced lactate production. It also increases cytoadherences, that is the ability of the red cell to get stuck to the wall of the microvessels. And it also disturbs autoregulation of blood flow and leading to hypoxia of distant tissue. And then this erythropoiesis, poor red cell developability, and anemia of many factors could occur from excess cytokine. I will explain red cell developability and anemia later on in the lecture. The role of cytokine in severe malaria also could cause problem of gluconeogenesis, hypoglycemia, loss of endothelial integrity, selective upregulation of vascular and intravascular adhesion, and as well as activation of leukocytes, platelets, thereby promoting procoagulant activity. Now, cytoadherences, which simply depict the stickiness of red cell, infected red cell to the wall of microvessels, and sequestration, which means the blockage of distal capillaries, thereby causing tissue hypoxia distant to that blockage. The rosetting formation simply connotes the setting around of a red cell, that is, a parasitized cell, red cell, attract more parasite red cell, increasing in size, thereby blocking the microvessels. So cytoadherence, rosetting formation, and sequestration are responsible for most of the tissue hypoxia that occur in cases of severe malaria. And you can see the effect of this on many vital organs, including the heart, the lungs, the brain, the liver, the kidneys, the intestine, and even the adipose tissue and subcutaneous tissue, and as well as the placenta during pregnancy. Now, the cytoadherence, what does it cause? The mechanism of cytoadherence and rosetting is what I will spend a little time to explain here. If falciparum infected red cell usually will bind to the post-capillary endothelial lining and to the non-infected RBC, both phenomena are thought to contribute to the occlusion of the blood flow and consequent severe diseases that it causes. Now, the parasite antigen, which stimulates interferon gamma and alpha, are released, and this upregulates the ICAM1, which is the main cytokine that is activated and promoting the cytoadherence further. And ICAM1 is suggested to mediate the parasite red cells ruling on the endothelium, while CD36 and CD31 and other receptors are responsible for more stable binding. Sequestration could be augmented by the ability of spontaneous rosetting, that is, the coming together of those red cells. Please note that infected erythrocytes often adhere to endothelium of the capillary and post-capillary venom, leading to obstruction, and then microcirculation becomes compromised. Now, there are other factors that contribute to the development of severe disease in severe malaria. If cytoadherence, rosetting, and sequestration formation of infected and uninfected occur in vital organs and it goes on uninhibited, the following could occur. It will ultimately block the blood flow, it will limit the local oxygen supply, hamper the mitochondrial ATP synthesis, and then stimulate the cytokine production. What about red cell rigidity and deformability, which is also a major mechanism by which falciparum infection induce the local tissue hypoxia that it causes? The outer red cell membrane becomes rigid once in parasitized red cell, and then become non-deformable. And please note that the maneuver of the red cell through the microcapillaries is because of the alteration in shape, because ordinarily the size of a normal red cell is almost the size of a capillary or even bigger in some instances. So the outer membrane rigidity and deformability contribute to the pathogenesis of severe malaria. So in patients with severe malaria, the entire red cell mass comprising most of the unparasitized red cell and also parasitized red cell become eventually rigid, partly because of several cytokines that also act on the membrane of the red cell, impeding and inhibiting the sodium-potassium ATP pump on the red cell, and also possibly because of the nitric oxide that is often released. So the mechanism of platelet endothelial cell interaction also contributes to the problem that occurred in cerebral malaria. In this situation, platelets, after activation by tumor necrosis factor, can markedly alter the function of the brain endothelial cell, either directly or by binding to the endothelium, or indirectly by releasing molecules from their secretory granules. For example, platelets release interleukin-1, which increases the expression of adhesion molecule such as intracellular adhesion molecule, that is ICAM1, and the production of cytokine such as interleukin-6 by endothelial cell. Platelet-derived microparticles also alter the production of cyclooxygenase, prostaglandin, and all of this might affect the endothelium. And the effects will include increment in adherence up to the leukocytes and parasitized red cell, increase in permeability and electrical resistance and apoptosis. Now, the increase in permeability causes fluid to move from intracapillary space into the extravascular space, thereby causing the edema that we often observe when during autopsy in cerebral malaria cases. Now, malaria-associated anemia is another major pathology that is due to severe falciparum malaria infection. So the severity of malaria-associated anemia tend to be related to the degree of parasitemia, because at the end of the day, both parasitized and unparasitized cells get destroyed because when there is overwhelming infection. So the pathogenesis could be due to several factors, and some of these include hemolysis or phagocytosis of parasitized red cell, ineffective erythropoiesis, phagocytosis of uninfected erythrocytes, autoimmune hemolytic anemia that is also being implicated, massive intravascular hemolysis leading to hemoglobinuria, and hemolysis after use of certain antimalarials. So these are the various mechanisms by which anemia occur in severe malaria cases. So there are a number of predisposing factors to complication of pre-falciparum malaria. These are as listed: 1. Children less than 5 years, pregnant women, immunocompromised or immunosuppressed individuals who have had splenectomy, lack of previous exposure to malaria, particularly those who live in non-endemic area, pre-existing organ failure, and internally displaced persons are all prone to severe forms of resistance. So I have put through this literature here to further help you understand the pathology and pathophysiology of malaria. Please lay your hands on them and read them, then listen to the slide as often as you can. I wish you good luck. Thank you for listening.